Okay.
Thank you so much.
Okay, so my name is Huang. So I want to tell you a little bit about the research on my lab. So my lab is entirely focused on Smith-Magenin syndrome, and we are focusing on a few different projects, including the neural function of RI1, as well as developing different kind of therapies for SMS using mouse models. So I want to tell you first about how do we understand the basic neurobiology of RI1. We are first focused on brain activity because SMS patients commonly have abnormal EEG brain activity patterns, and some patients have clinical seizures. And this is a problem because seizure is a hyperactivity of the brain, if you will, and too much activity will maybe sometimes damage your brain, and then it will impair the quality of life. And one of the problems to understand how RI1 regulates brain activity is that, at least in mice, and also that's true in human, RI1 is expressed throughout the brain. It's very widely expressed. So how do we identify brain regions important to seizures, right? Like what brain region is responsible when you have RI1 loss to drive neural hyperactivity? So to do this, we use a technique called brain clearing. Basically, as you can see here, this is the before and after clearing. We can basically clear the whole mouse brain to remove all the unnecessary lipids and do whole brain imaging and immunostaining. So we clear the mouse brain, and then we stain for one protein called FOS, which is a marker for brain activity. So if a brain region has too much FOS protein, that means that that brain region has a lot of activity. And after whole brain imaging, because the mouse brain is now clear, we can look at the whole brain. We can identify brain regions with too much FOS signals. So here's an example of the FOS signal. So I want to focus on this region of the mouse brain called hippocampus. And we found that in RI1 mutant mice, there are too much FOS protein if you use a kainic acid treatment, which is a drug to increase brain activity. This brain region will show too much FOS activity. So that suggests that this brain region could be hyper-excitable in the SMS mice. So to look at this problem in another way, we put electrodes in the mouse brain and record the brain activity, and we can see that our mutant mice, they spend more time in seizure. So this is a light mouse, right? So this is another evidence suggesting that losing RI1 in the hippocampus, so the electrodes are actually put into the hippocampus, have too much activity. And then we can also do another way by taking the cells out of the mouse. We can prepare acute brain slices and basically use a pipette to listen to the neurons talk, if you will. So this graph shows you that in the control mice, there's some activity in the neurons coming from the surrounding neurons, the neighbors. And in RI1 knockout mice, the neighbors talks a lot, right? There's more activity from the neighbors. And how about RI mutant neurons themselves in the dentate gyrus, hippocampal dentate gyrus neurons? We can see that the RI mutant neuron themselves also talks a lot, very frequently. So this kind of hyperactivity could be contributing to the susceptibility to seizure in SMS mice as well as patients. So what happened to these neurons, right? So we analyzed gene expression and found that there are upregulation and downregulation of some ion channel proteins. These are the proteins that allow the flow of ions in and out of the cells. So for example, too much of this protein will allow too much calcium ion coming into the cells and potentially make the cells more excitable. So we tested by adding a drug called NNC55 to inhibit this ion channel. And as a result, we can see that when the RI mutant neuron, they talk too much, but if you apply this drug to block this ion channel to reduce the activity of this channel, then the neurons will become quiet down. So we are looking into this and to see if we can further understand what's going on here. But this is just one example of how we identify the important brain regions in the SMS mouse brain and then understand what are the molecular and cellular mechanism that potentially contributes to seizure in the mice. So now to gene therapy project. As you can remember from the video, every patient has a good copy of RI1, right? So another way is how do we increase the expression of this good copy to treat SMS? So we're using a technology called CRISPR activation system. Basically, they have two components. One component is the RNA component. Basically, this molecule here will direct another component, the protein component, to the RI1 gene. So this is the remaining good RNA copy. We want to find out a way to enhance the expression of this copy. So with the protein component and the RNA component together, this protein can activate the expression of the nearby gene. In this case, because this RNA is located upstream of RI1, so the protein can activate RI1. So this would be the concept of this design. So basically now we found we identified different RNA that can bind to the upstream of RI1 gene, trying to increase RI1 expression. And we found a sequence, RNA SG2. We found that it can increase actually RI1 expression by 2-fold. So this is promising. And then we make adeno-associated virus, AAV, which is the gold standard for gene therapy right now. So we replace all the toxic genes and then put in the necessary component, the protein component and the RNA component I talk about. We made a virus out of it, and then we treated neurons with this AAV virus to see if we can increase RI1 level or not. So this is a wild type mice, right? You use it as the one. And the SMS mice, the neuron have few lower RI expression. And we found that with the treatment of this AAV, we can increase RI1 expression level. So that is very encouraging. Then we injected this virus, the treatment virus, into mice. So we selected a brain region in the hypothalamus called PVH. So this brain region, in our recent work, we found that it's important to mediate obesity in the SMS mice. So we thought this would be a good place to start. So we basically generated wild-type mice with control virus, SMS mice with control virus, and SMS mice with the treatment. So the purple one is a treatment group. One of the features of SMS mice is they have a lot of repetitive behaviors, and you can see here in red. But after treatment, you can decrease the repetitive behavior, as you can see here in purple. Another feature of SMS mice is the increased body weight, right? Compared to control in black, SMS mice have eventually gain body weight, as you can see in red. And after the virus treatment, we found that although the mice eventually still gain weight, their body weight is much lower than the SMS mice receive control virus treatment, and the onset of their obesity is also much delayed. So this suggesting that the good copy of RI1 has some effect. And to remind you, we treated the mice at about postnatal 3 weeks of age. So it is like an early adolescent equivalent to humans. So to summarize the second part of our project, I told you we used a novel CRISPR AAV system that have a protein component and the RNA component to increase RI1 level. And we can inject the virus that encodes this component into the mouse brain of SMS mice to increase RI expression and partially rescue some of the obesity and stereotypic repetitive behavior. So we can understand the remaining good copy of RI1 can modify, but if you increase the expression, it can modify disease progression. So the final part of the project, also the part I'm more excited about, is developing a BDNF therapy for SMS. So RI1, our previous work found that it's a protein that binds to DNA and then upregulate or downregulate a lot of different target genes. And one of the important target genes of RI1 is called brain-derived neurotrophic factor, or BDNF. And SMS, because they have fewer RI1, so it have decreased BDNF expression in the hypothalamus region. So what BDNF does is that it's a secreted molecule. It binds to the downstream receptor of surrounding cells to activate downstream pathway that promotes neuronal health. So if you don't have this BDNF, you have fewer of this BDNF, you have fewer of these signaling pathways. So as a result, neurons might not be as healthy. So our first experiment, we figured, why don't we put human BDNF back into the mice. So this is the human BDNF transgenic mice that basically express human BDNF from early development. So we want to ask, what if the SMS mice develop with the normal amount of BDNF? So you can see here in blue is a treatment group. Again, SMS mice is the red one. BDNF level is increased with the human BDNF expression. And then the obesity you see in the red group, the SMS group, is gone in the SMS mice treated basically genetically expressed human BDNF. And the body weight remains similar to control mice, even when they are very old. So that means that if you have BDNF expression in early in life, you can prevent the development of obesity. So HDL, LDL, and leptin are some of the protein and lipid that is associated with obesity. And they are dramatically increased in SMS mice, but they're also decreased when you have human BDNF over expression. So these are the section of the adipocyte of fat cells. So in SMS mice, the fat cells are quite big and large. They accumulate a lot. But if you have a BDNF treatment, again, they look very similar to control mice, suggesting that the size of the adipocyte can be decreased by this genetically expressed human BDNF in mice. So another defect we can see in SMS mice is that they are more shy, if you will. So there's an assay, if you put two mice on the different end of the tube and you let them meet in the middle. And what happens most of the time is in control mice, they will push forward. They are very interested in this kind of social engagement to the extent that SMS mouse usually will just retreat and then leave, sort of losing the match. And what happens here is SMS mice will basically retreat. They are more shy. They leave the tube 95% of the time. But with human BDNF expression, they behave more like a control mice. So they win 50-50% of the time. And when they encounter other SMS mice, they become less shy, right? So they stay in the tube more. They engage in this kind of face-to-face social engagement more than the SMS mice, suggesting that expressing of human BDNF can also modify disease progression in terms of social interaction. So this is all sound very nice, but this is a genetically expressed molecule from early development. But can we figure out a way to use pharmacological approach and to increase BDNF signaling? And especially we are interested in adult mice, because I think in treating adult mice is maybe having more impact in the human patient population. So just a reminder, right? BDNF is a secreted molecule. It has to bind to TrkB receptor to enhance downstream signaling. And we found a drug called LM22A4. It is a small molecule that mimics the BDNF domain. So basically, it can also enhance the TrkB signaling pathway. It has been shown to have some potential in other disease. And we are using this drug to see if we can reverse or delay some of the symptom progression in adult SMS mice. So this project will be the main focus of our lab next year. But we do have some interesting preliminary results showing here. So we have very few number of mice. So here in orange is a control mice treated with saline daily, and we have 3 experimental SMS mice treated with this drug I just talked about, also daily. So we have 3 of them. And when we treat them at 8 weeks of age, so they are considered adult. So basically, we can see that the body weight of these mice treated with drug decreased dramatically compared to control. And we isolated their fat from liver or other organs. We found that in the control SMS in gray, they have increased fat mass, but with the drug, we can sort of decrease these fat mass. So the goal for next year is to use this drug and then analyze if this drug can rescue obesity and social interaction and repetitive behavior in SMS mice. So to summarize my talk, I show you we studied the basic neural function of RIA1, and we identified that RIA1 loss causes a hyperexcitability of hippocampal neuron in the brain that could potentially facilitate seizure. And we also have a drug that can decrease the hyperexcitability of these neurons. I show you that using CRISPR activation gene therapy, we can increase RA1 level in mice and then partially mitigate some of the disease feature. We show you that the genetic expression of human BDNF can prevent the development of obesity and social interaction defect. And finally, for next year, we will test in pharmacological treatment targeting the BDNF pathway to enhance it, can reverse or halt the disease progression in adult SMS mice. So I'd like to thank my whole lab, and then I'd like to thank SMS Research Foundation for supporting our research, right? I started my lab in the middle of the pandemic, so it's quite difficult. It is actually impossible without the support of SMS Research Foundation. So we are really grateful for that. If you have any question, you can email me. Here is my email address. Thank you.
Excellent. Excellent. Thank you, Hsiang. This is I would suggest to actually start with a few questions after each of the presentation briefly, so we can move to and then move to the next presenter. There is a Q&A function below, so any of the participants feel free to write any questions you may have within the Q&A function. If you click on it, you can actually write the question. I'm going to scan through them for each of the presenters in case there's any. As you are thinking about potential questions, Hsiang, I have a couple of questions for you. So obviously BDNF affects the obesity, as you mentioned, but also the neuronal compartmental model. What do you think that, how do you think that the drug in BDNF will actually be able to affect the neurological manifestation of the disease? So because it's obviously more downstream than RAI, are we missing some of the key potential function of RAI, and what is your interpretation of that?
Yeah, so based on our genetic study of overexpressing BDNF genetically, it does treat the obesity and the metabolic dysfunction associated with obesity, but surprisingly, it can also improve some of the neural functions such as social interaction, right? So it goes beyond just the obesity. So that's why in our drug trial, we are going to test all of the basically defect, behavioral or obesity we can measure in SMS mice.
Okay, thank you for the answer. And also the other point I wanted to ask you, you're now going to test at least the therapeutic intervention in adult mice, which is absolutely exciting because unfortunately there's always a misconception that SMS can only be treated as children. And actually we don't know that. There's a higher probability when you're a child probably to be treated, but it's not impossible that you may do an effect also in adults. So I really look forward to seeing your data in adult mice as well. One simple question, are you going also to assess though the effect in younger mice, either adolescents or newborns, to see if the impact on the drug may even be more substantial and more effective in a way?
So right now we are focusing on adult because these drugs are expensive and we have a limited amount of resources and everything. So we want to focus on kind of the most difficult thing, because if we can treat adult mice, then we have more reason to. We will be very optimistic to treat even younger mice. So that is the one thing we're focusing on right now. But eventually, we can focus on different ages and see if there is any defect, right? If there are some features cannot be rescued at adult, we can push forward younger and to see if we can treat the features in younger mice for sure.
Very good. So we got a question online from Richard Cartwright. So I'm going to ask you that question. Is there any known negative effect if the BDNF is overactivated or excessive?
So too much BDNF, I think, is also a tricky thing. So one thing we noticed was that if you had too much BDNF in wild-type mice, so remember the genetic overexpression of human BDNF in mice, we also overexpress that in mice. And as it turned out, if mice have overexpressed BDNF, their body weight is basically what we saw was fine. The adipocyte size was similar to control, but the only thing we found weird was the social interaction. They actually become more shy. So it looks like the too much or too little BDNF can modify the social interaction in wild-type mice as well.
Thank you, Hsiang. And maybe just to add to that, I would say that one of the advantages of having a molecule, a small molecule as an approach, is that you can modulate the dose and to be able also to assess the level of activation of BDNF. So that could be one of certainly elements that will be assessed, of course, in mice, and then potentially, if one day we'll be able to in human as well.